Thomas, at the beginning in your presentation, you were alluding to, and some of the other titles sort of alluded to the fact that it's a shame that this hasn't gotten the clinical traction that many of us think it should have gotten. And I'm going to tell you why I think that's important. So nowadays, a patient presents to a U.S. oncologist, and the first thing that the oncologist, if this patient has either never been treated, or has already been treated and the cancer has either not responded or has recurred, what is the oncologist most likely to say, or what are colleagues most likely to ask them? They'll send it to Foundation, and I'll explain what that is in a second. Or sometimes now we send it to CARES or send it to Tempus. Foundation One is basically an organization, a for-profit business that will sequence your DNA from the tumor that your patient has. Actually, what it does is it doesn't sequence the entire genome. It's not whole genome sequencing. It takes over 300 genes. It's actually currently 309 genes, 309 genes that we all think are important in cancer. And then additionally, 36 genes that are involved in rearrangements, and I'll come to that later, and basically gives you a report back on that. What are those 309 genes? They're shown here, and there'll be a quiz on this at the end of the talk. No, I'm just trying to point out to you, by the time you have 309 things on a slide, they get to be very small. But they're the kind of genes that we all sort of associate with cancer. P53 is here, KRAS is here, BCL2 is here, AXL is here, BRCA is here. So genes that we associate with cancer are in here. And then some years ago, after it had been started, they realized that, well, not all genes that are involved in cancer need necessarily mutated. In some cases, we'll have rearrangements that occur. So there are a set of 30 some odd genes, 35, that they look for rearrangements with the promoter of another gene and the critical portion of that gene. And in this context, I'll point out NTRK1 and NTRK2 here, which are genes that get sequenced in everybody and which get assayed in everybody. And they occur overall in about 1% of tumors. So that's how deep we're diving into this. We're willing to spend time, money, effort looking for a gene that is responsible for some role in only less than 1% of cancers. The output from that is that it basically classified mutations as category 1, 2, or 3. And basically, category 1 are mutations that we know are important in cancer and for which we have a drug. And ideally, for which we have a drug that has been shown to work in your cancer. Because, for example, if you have mutation in the EGFR and the epidermal growth factor receptor, and you have lung cancer, we've got really good drugs for tumors that have that mutation and have lung cancer. If you see that same mutation in, for example, a gastric cancer or a colon cancer, those drugs don't work. So there's something about the mutation and the tumor in which it occurs. So category 1 are those. Category 2 are, well, this might be important, but we don't have a drug. And category 3 is really, I don't know. It might be potentially important, but we don't have enough information yet. So what it basically does, it gives you a report. And what you see here, for example, and you'll see there that it says ALK rearrangements, which is something, again, that we see in lung cancer, and that's where they're important, not anywhere else. Although the ALK stands for anaplastic lymphoma kinase, where it's really been shown to be important clinically, is actually in non-small cell lung cancer. And there's 4 drugs that are currently approved for the treatment of ALK, and it gives you those 4 drugs as choices. And so they have a long list according to the cancer. Here's non-small cell lung cancer, melanoma, breast cancer, colorectal cancer, ovarian cancer, cholangiocarcinoma, prostate cancer, and solid tumors in general. And so they'll give you recommendations. But what you need to know is that the recommendations that they give exceed that for which they have FDA approval. And you need to know that the doctors use this, although there is really no FDA verification that this is important. So what's the problem? In the United States in 2022, it is expected that 609,360 Americans will die of cancer. And it's not because their tumors did not have, quote, actionable mutations, meaning mutations for which we have drugs, but because their tumors are, in fact, drug resistance. Everybody who dies of cancer dies with a drug-resistant tumor. This is a list that I stole. So this is the list of the genes in Foundation that are evaluated. And there's very few of the genes that we consider to be drug resistance genes. This is a list that I stole in part from Michael Gottesman, and it's a list of genes that confer drug resistance. Only a handful of these are found in the Foundation 1 list. The reason is that they primarily confer drug resistance, not by acquiring mutations, but by overexpression or underexpression. Looking for mutations in these genes is unlikely to provide answers that let us understand why tumors fail to respond and why patients die. So we know that it's expression. So since this is about ABC transporters, Susan and I worked on this long ago, and this was in the mid-1990s. What we did, we looked at the expression of genes in the NCI anti-cancer drug screen, which was 60 cell lines. And basically the way the data was plotted at that time, and still is you would get for any drug, basically a profile that would show you how active it was among the 60 cell lines. And the central point was the median of all that activity. So you were either to the right or the left of that median. What we did was we looked at the expression of PGP in cell lines, and we were able to correlate lack of activity, shown here as these bars coming over to the left, with high levels of expression of MDR1. In other words, expression of drug-resistant genes was what mediated drug resistance. We identified in this way nearly 500 substrates amongst those NCI cancer drug screen drugs that have been screened in the NCI cancer drug screen, and the majority of those were indeed P-glycoprotein substrates. So then why do we do this? Why do we send our list to Foundation if we're not getting all the information? And indeed, what I would argue, the important information. The reason is because this is difficult. So what you here see is an expression array. It's difficult to interpret. It's much easier to look for mutations than to look for changes in expression. And then that's why we end up doing this. So what about the rearrangements? Well, we look for those, but again, we're not looking for rearrangements that confer resistance. And in ovarian cancer, there's a large group of individuals not long ago reported that they had found the rearrangements involving P-glycoprotein and a variety of genes in ovarian cancer. Therein lies the problem. The genes that were linking to P-glycoprotein and upregulated it varied across the different cancers. And searching for this would be very difficult. Foundation looks for rearrangements that are very well defined. By the way, in this study, which I was finally alerted to by Pete Borse, who wrote me an email one morning, and all that it said was finally, and I knew what he was talking about. They talked about gene rearrangements as the cause of resistance. They failed to overlook the fact that 15 years earlier, we had reported this, and that had they done a simple Google search for MDR1 rearrangement, at the top of that Google search out of 102,000 results would have been our publications. We were able to show this even in samples from patients with leukemia. These were children with leukemia, and what you see here is probes that were looking at the rearranged genes, both the P-glycoprotein and the now important promoter of another gene that was upregulating the expression. So none of these individuals failed to find that in the literature. And it always reminds me of what Dan Sackett, who used to work with us, and we wish he still did, but we now at Columbia and he's at the NIH, used to say, an hour at the library can save you a year in the laboratory. So is this important? In fact, it is. So Dr. Bates and I have analyzed the growth and regression of clinical data from over 140,000 patients. And what you can do is you can take the actual growth and regression of tumors, which is what you see normally when you're treating cancers, an initial regression and then a subsequent growth. And you can basically break that down into a fraction of tumor that is regressing because it's sensitive to the drug, and a fraction of tumor that is growing because it's resistant to the drug. There's absolutely no correlation between regression of the tumor and the patient's overall survival. But growth of the drug-resistant tumor is what impacts the patient's overall survival. And we can do this because when we look at the rate of death and we compare it to the rate of growth, we can see across all cancers that the rate of growth, which we designate G, is indeed an excellent biomarker of overall survival. What you're seeing here is we took over 800 patients with non-small lung cancer. We determined the rate of growth of their tumor, and then we looked to see what the rate of overall survival was. This is the best overall survival. This is the worst. This was the quartile of the data with the slowest rate of growth, and this was the quartile with the fastest rate of growth. This is true across all cancers, across 140,000 patients. So it is growth of the drug-resistant tumor that is responsible for why patients die. And unfortunately, we have no insight into what those genes are in any patients and whether or not their tumor have that, and we should be aware. So in summary, we test extensively, looking for what alterations in a tumor might lead to a therapeutic response. But patients eventually die of drug-resistant tumors. And in the overwhelming majority, that drug-resistant clone was likely there to begin with. It just grew out over time. It would be nice if we knew that. Eventually, having that knowledge will help us be better doctors, but that will be much more difficult than looking for simple alterations such as mutations, for which you can do this quickly and without a whole lot of problem and charge a lot of money. We will need expression data and knowledge of what level of expression is low enough to overcome and that we can ignore, and what level of expression is high and will prove lethal because it will prevent drugs from working. We've done the easy part, look for mutations. Now we need to think how we do the hard part. So with that, I'll end my slide. I'll stop sharing, and I'll be happy to take any questions.
Thanks a lot, Tito, for a very thought-provoking presentation and worrisome. I mean, so what to do if we're not testing? I mean, the test should be available. I mean,
right. I mean, you know how difficult it is to express, to look at, cDNA arrays and that data, and interpret them, right? And then the problem is what we've always known in P-glycoprotein, we always say, is that level of expression important? Is it significant to confer resistance? So we have to come up with values for that. And we probably would have to have values for different cancer, because possibly a little bit of P-glycoprotein in one tumor might be enough to confer resistance, but in another tumor, you may need larger amounts. And you may need larger amounts for some drugs than you do for others. It's a complicated thing, In the laboratory, we can do this, but we've got to figure out how we do that with patients.
Right. And it could be a double-edged sword, as Michael might comment with his question. Michael?
So, no, first of all, Tito, thank you for helping us think really important thoughts about the issue of treating drug-resistant cancers. You probably know that one of the things that we're doing now in my lab is we're trying to create a larger catalog of genes that are important in drug resistance. We know the ABC transporters are, but there are clearly, as you pointed out, many, many others. Do you think it's possible? I mean, I think we're doing these CRISPR analyses. We activate, we inactivate, we knock out genes. We're getting a lot of genes that get in tissue culture with resistance to specific anti-cancer drugs. It's going to be hundreds. It's probably going to be many more resistance genes, and we know that already, than there are oncogenes. Is this a solvable problem? Is it going to depend on there being major players and minor players, and so you can deal with most drug-resistant cancers, but not all? Or can we actually figure out how to solve this problem?
I mean, I think we can figure it out. But then the question is, will people be interested in what we figured out?
They haven't been so far, I guess.
Right. Yeah. So what happens is, you get an EGFR mutation and, oh yeah, we got a drug for that can be used and that a company can sell it, right? So it would have to be in a situation where you have more than one option, which is increasingly common, okay? And I have drug A, B, and C. And if a mechanism of resistance that you identify is going to impact drug A, that then becomes important because you have B and C as a choice, and you won't use A and you'll use B and C. So I think that yes, we're heading in that direction because we have an increasing number of options for cancers. And you sit there and you say, which one am I going to use? I'm going to throw A or B or C, like in prostate cancer. There's a plethora of options now in first line and second line and third line. We need guidance. And the majority of these aren't mutation directed, but they clearly are mechanisms of resistance. And if they can be identified, then it can inform a doctor's decision.
So one big advance, I think, is that the FDA now requires that when a new drug comes on the market, that there be some description of how it's handled metabolically and by transporters like P-glycoprotein. So that's sort of encouraging some of the companies who are making anti-cancer drugs to shy away from known PGP substrates or known G2 substrates. What I'm wondering, though, is whether or not, and then maybe a great example of that is you mentioned ALK positive non-small cell lung cancer. That's maybe 5% of lung cancer. And the original drug, ceritinib, which was used to treat it, is an incredibly good PGP substrate. And when you look at the tumors, a lot of them express high levels of PGP when they're resistant. But the second and third generation drugs are not very good PGP substrates. So that maybe that's an example which took industry and oncologists a long time to learn that this is something was pretty much figured out up front and saved a lot of development time. Maybe not made as much money. That's the problem.
Right, exactly. But no, Michael, that's a perfect example because they're still out there. And maybe you don't tolerate B and C, and you have to go back to A, the original one. So, which was the one that was a strong PGP substrate. But if you knew that the patient's tumor doesn't have PGP, okay, and then you also know that they don't have brain metastasis because that's where the non-PGP substrates were actually more effective in getting into the brain and treating the brain metastases that were there. But if you knew that both of those things, then if A is tolerable, you have an option. But so, yeah, there's already examples of where knowing that would have been of value. And so what now needs is a little bit of financial incentive to drive that. And maybe as a community, what we ought to do is say, okay, which are the ones that are, if we know that they're drug resistance genes, that they're going to confer resistance, and look, forget whether the level is 10, 20, or 100. All of those are bad. So that we don't have to factor in that, is this level important or not? Just any level is important, okay? And try to push forward a few of those as genes that we should start to be considering. So we ought to talk more, Michael. We ought to think about what drugs are available, where our choices are available, and do we know some drug resistance genes that would inform the decision? I think we can come up with some good suggestions. And then one has to conduct an experimental trial, maybe even in the real world, where you'll check for expression of that in the tumor or not, and let that inform your drug decision and see how well that does.
There's another element. You mentioned that it's likely that many of these mechanisms of resistance pre-exist in heterogeneous tumor populations at the time of presentation. But let's say a fair amount of resistance evolves during treatment. We need a way of, and you know that physicians and IRBs are very reluctant to allow you to continuously biopsy solid tumors during the course of treatment. Right. So you do need a way of detecting maybe circulating.
The emergence. Yeah, and also, yeah, right. But, circulating, they're doing that now for DNA. But boy, circulating RNA, that's going to be really challenging.
Oh, because it doesn't.
But by the way, IRBs and all these people, but there's a lot of patients that are very motivated. If you tell them, if I biopsy your tumor, I might get meaningful information, they'll say, do it. Sign me up. Can you do it right now? Yeah. Or at the latest tomorrow? So I think, we just have to we have to think hard about it and see where we, model this, where we bring it to people and say, look, this is meaningful. Let's see. So I'll reach out to you.
Yeah, let's talk.
Okay, all righty.
So that was great
Okay,
having you guys discuss this very important topic.